Welcome to the Life Science Coach. Today's topic is fish tagging methods, an important technique used by scientists to track fish movement, study populations, and support marine conservation. In this video, we'll explore different tagging methods and how they help researchers understand aquatic life better. Like, share, subscribe to the Life Science Coach. Why do scientists tag or mark fish? Fish tagging is one of the most important techniques used in fisheries biology and marine research. By marking fish, scientists can track their movement and migration patterns across rivers, lakes, and oceans. These methods also help researchers estimate fish population size, study fish growth, and understand natural mortality rates. Fish tagging provides valuable data that supports conservation, sustainable fisheries management, and aquatic ecosystem research. In simple words, tagging helps us better understand underwater life and protect marine biodiversity for the future. Fish tagging methods are mainly divided into three major types: biological, chemical, and physical tagging methods. In this section, we'll focus on biological or natural marking. Biological tagging uses natural characteristics present in fish for identification and tracking. These natural marks can include parasitic marks, morphological marks, genetic marks. Parasitic marks involve identifying fish through specific parasite patterns. Morphological marks are based on body shape, color patterns, fins, or natural body structures. Genetic marks use DNA and hereditary traits to distinguish fish populations and species. These methods help scientists study fish populations, migration, biodiversity, and conservation more effectively. Now let's understand the different types of biological or natural fish marks. The first type is parasitic marks. These are used to identify stocked fish and study their movement or migration patterns. The major advantage is that they are natural and have low survey costs, especially useful in large water bodies. However, trained personnel are required, and mathematical errors may sometimes occur. Next are morphological marks. These marks help scientists differentiate fish stocks and are widely used in mark recapture studies. Their main advantage is low cost, but environmental conditions can change physical characteristics, making identification difficult. Examples include meristic counts, pigmentation, body shape and size, age marks of otoliths or scales. The third type is genetic marks. These are highly effective for identifying fish stocks using genetic characteristics and DNA-based methods. Although very accurate, they can be expensive because they require special reagents, equipment, and proper blood sample preservation. Common examples include heavy checkmark electrophoresis, heavy checkmark serological methods. Another important fish tagging technique called chemical marking. Chemical marking involves the use of specific chemicals or dyes to mark fish for identification and research purposes. This method has several advantages: low cost, reduced handling time, large numbers of fish can be marked at once, even smaller fish species can be tagged effectively. However, one major disadvantage is that individual organisms may not always be recognized separately. Some commonly used chemicals include oxytetracycline or OTC. This method is less costly and produces reliable marks, but it takes time for the mark to develop, and special equipment is needed to identify the marks. Alizarin complex 1 or ALC. This chemical has a low mortality rate and is considered an effective way to mark fish. The main disadvantage is the high cost of the chemical. Calcium is another chemical marker, but it is sometimes less reliable in producing clear marks and may also result in higher mortality rates. Chemical marking methods are widely used in fisheries science for population studies, stock assessment, and conservation research. Let's look at the different methods used in chemical marking of fish. There are three major methods: immersion, injecting, feeding. In this slide, we'll focus on the immersion method. In immersion marking, fish are placed in water containing vaccine antigens or chemical marking agents. The fish absorb these chemicals through a dip or bath treatment, helping create identifiable marks while also increasing protection against diseases. This method is especially useful for marking large numbers of fish quickly and efficiently. Here we can see rainbow trout submerged in an oxytetracycline chemical bath, a commonly used immersion marking technique in aquaculture and fisheries research. The immersion method is simple, economical, and widely used for population studies and fish health management. Two important methods of chemical marking in fish: injection and feeding. Injection is a simple and direct technique where the chemical marker is injected into the fish body. However, proper concentration of the substance and the correct injection site are very important. Incorrect dosage or location may affect the marking efficiency and fish health. The second method is feeding. In this method, fish consume food mixed with chemical dyes or markers. Feeding works especially well in laboratory and hatchery conditions where fish can be closely monitored. But if monitoring is not done properly, some fish may consume less feed than others, resulting in unclear or uneven marking. Physical tagging mainly involves the use of tags or body modifications for fish identification and tracking. These methods are broadly divided into internal tagging and external tagging. External tags are attached to the outer surface of the fish body, fins, or muscles. Some common external tags include Carlin tags, anchor tags, spaghetti tags, dart tags, and fin clips. These tags are easy to identify and are widely used in fisheries management, migration studies, and population estimation. Internal tagging involves placing the tag inside the fish body cavity. Examples include electronic tags, telemetry tags, and passive integrated transponder or PIT tags. Internal tags are useful for long-term tracking because they are protected inside the fish body and reduce tag loss. Physical tagging methods are highly durable and effective for studying fish movement, growth, migration patterns, and stock assessment. Internal tagging involves placing tags inside the fish body for long-term identification and tracking. There are several important internal tagging methods used in fisheries research. These include body cavity tags, coated wire tags, radio tags, fish positioning sounders, subcutaneous tags, micro tags, DST GPS fish tags, Simrad GPS sonar, and passive integrated transponder tags, also known as PIT tags. Among these, body cavity tags are commonly used. In this method, small metal anchors or tags are placed inside the body cavity of the fish for later identification. These tags can later be recovered and detected using strong magnets or specialized detection equipment. However, there are some limitations. If the tags are inserted before spawning, fish may naturally expel the tags. In some cases, machinery or handling processes may also damage the tags before they are detected. Internal tagging methods are highly useful for long-term monitoring, migration studies, fish stock assessment, and fisheries management because the tags remain protected inside the fish body. The first type is subcutaneous tags. These are inexpensive plastic discs printed with serial numbers and implanted between the outer skin layer and muscles through a small incision. They are commonly used for identification because they are simple, economical, and easy to detect. The second type is coated wire tags. In this method, a very small wire tag is injected into the fish using special applicators or by hand. These tags can be inserted into different body regions such as the snout, neck, caudal fin, or muscular areas. Coated wire tags are detected using sensitive metal detectors or X-ray equipment. Another important method is the passive integrated transponder tag, commonly called the PIT tag. A PIT tag is a radio frequency device that transmits a unique identification code to a reader. The code is displayed in numeric or alphanumeric form for accurate fish identification. PIT tags are highly useful for positive identification and fisheries research. The first method is microtags. Microtags are microscopic plastic chips made up of multiple colored plastic layers. These layers can be color-coded to identify different groups of fish, similar to coated wire tags. One major advantage is the huge number of color combinations available, allowing researchers to identify millions of unique fish groups accurately. Next are radio tags. Radio tags transmit radio signals through antennas to a receiver. These systems are most effective in shallow and low-conductivity water environments. The receiving antennas can be mounted on airplanes, boats, snowmobiles, portable towers, or even carried manually along streams and rivers. Radio tagging is highly useful for studying fish movement, migration patterns, and habitat behavior in real time. Another important method is sonar tags. Sonar tags are hydrostatic tags in which identification details and reporting instructions are enclosed inside a plastic capsule attached to the fish. These tags help researchers identify individual fish or fish groups during tracking and monitoring studies. Advanced tagging methods like microtags, radio tags, and sonar tags provide highly accurate data for fisheries management, migration analysis, stock assessment, and aquatic conservation research. DST stands for Data Storage Tag. These GPS-based tags can either be attached externally to the fish or implanted inside the body. The main function of DST GPS tags is to collect and store information about the fish's geographic position and movement patterns. These tags communicate with satellites and onboard SIMRAD sonar systems installed on research vessels. Using this technology, scientists can accurately track fish migration routes, swimming behavior, habitat usage, and environmental conditions. Fish positioning sounders and SIMRAD GPS sonar systems help researchers receive and analyze movement data in real time. These technologies are highly important in modern fisheries science because they support stock assessment, fisheries management, marine conservation, and ecological research. External tagging methods involve attaching tags to the outer body surface of the fish for easy identification and monitoring. These methods are widely used in fisheries research, stock assessment, migration studies, and conservation programs. There are several important types of external tags. The first method is mutilation, where specific body parts such as fins are clipped or modified for identification purposes. Another method is Peterson discs, which are disc-shaped tags attached externally to the fish body. Strap tags and Carlin tags are commonly used for long-term identification because they are durable and easy to recognize. Dart tags and spaghetti tags are also popular external tagging methods used for tracking fish movement and recapture studies. Other methods include branding, paper fasteners, dangler tags, and anchor tags, also called Floyd tags. These tags are attached externally and contain identification numbers or codes for tracking individual fish. Another important technique is visible implant elastomer, or VIE. In this method, colored elastomer material is injected beneath transparent tissue areas, creating visible identification marks. Let's look at one of the simplest external tagging methods called mutilation. In this method, fins or other body parts of the fish are clipped, punched, or removed for identification purposes. These marks help researchers identify individual fish later during recapture or monitoring studies. A common example is the removal of the adipose fin, as shown in the figure. Mutilation tagging is inexpensive, easy to perform, and useful when tagging large numbers of fish. However, there are important disadvantages. Removing or damaging body parts may negatively affect the fish's behavior, swimming ability, growth, or survival. Because of these limitations, mutilation methods are used carefully in fisheries research and management programs. Even though it is a simple technique, it has played an important role in fish identification and population studies for many years. Branding and visible implant elastomer, also known as VIE tagging. Branding is a method where hot or cold instruments are applied to the fish body to create a recognizable mark for identification. These marks help researchers identify fish during monitoring and recapture studies. Hot branding uses heated instruments, while cold branding uses extremely low temperatures to produce visible marks on the fish body. Another important method is visible implant elastomer or VIE tagging. In this technique, liquid elastomer tags are injected beneath transparent tissues of the fish. After injection, the material quickly cures into a soft, biocompatible solid and remains externally visible. These tags are commonly placed near transparent areas such as around the eyes or fins for easy identification. VIE tags are highly useful because they are safe, flexible, and allow individual fish recognition without causing major injury. Peterson discs are round disc-shaped tags that are inserted beneath the dorsal fin of the fish using a pin. These tags remain externally visible and help researchers identify individual fish during monitoring and recapture studies. Peterson disc tags are widely used in fisheries research because they are durable and provide reliable identification over long periods. They are especially useful for studying fish migration, growth, population dynamics, and stock assessment. However, one important disadvantage of this technique is the long application time. Attaching the disc carefully beneath the dorsal fin requires precision and handling, making the tagging process slower compared to some other methods. Paper fasteners are used similarly to strap tags for short-term marking studies. These tags work best on fish that have strong and bony opercles, which help hold the tag securely in place. Because they are simple and inexpensive, paper fasteners are useful for temporary identification and short-duration fisheries research. The second method is strap tags. Strap tags are made of lightweight, non-corrosive metal, and are available in different sizes. Each tag is usually serially numbered for easy identification. These tags can be attached to different parts of the fish body such as the opercles, fins, or caudal peduncle. Strap tags are durable and commonly used in fisheries monitoring, migration tracking, and population studies. The third method is dangler tags. Dangler tags consist of a tag carrying identification information attached to the fish using wire. When these tags are applied close to the body and designed in a streamlined shape, they are retained very effectively and may remain attached to the fish for its entire life. These external tagging methods help scientists identify, track, and study fish populations more efficiently in both freshwater and marine environments. The first method is the Carlin tag. A smaller version of this tag, known as the fingerling tag, was specially developed for small and juvenile fish. The Carlin tag consists of a plastic disc attached to the fish body using stainless steel wire or polythene thread. These tags are durable and commonly used in fisheries research for long-term identification and tracking studies. Carlin tags are especially useful in migration studies, growth monitoring, and stock assessment programs. The second method is the spaghetti tag. A spaghetti tag is made of flexible vinyl tubing that passes through the fish body using a hollow needle. Identification information or serial numbers are printed directly on the tubing for easy recognition. These tags are lightweight, easy to apply, and highly visible during recapture studies. Spaghetti tags are commonly used in marine fisheries research because they allow quick identification of fish in the field. The first method is the dart tag. Dart tags are made of a nylon shaft with a barbed end and a vinyl tube fitted over the upper part of the shaft. The barbed end helps secure the tag firmly inside the fish body, while the vinyl tube contains the identification information. These tags are commonly used in fisheries research for tracking fish movement, migration, and population studies. Another important method is the anchor tag or Floyd tag. This tag is actually a modified version of the dart tag. In anchor tags, a nylon T-bar replaces the harpoon-like head found in traditional dart tags. These tags are inserted using a special tagging gun, which can be loaded with one or multiple anchor tags for rapid tagging operations. Anchor tags are highly visible, durable, and widely used in both freshwater and marine fisheries research. In this video, we explore different biological, chemical, physical, internal, and external fish tagging techniques used in fisheries science and aquatic research. These tagging methods help scientists study fish migration, growth, population dynamics, stock assessment, and conservation management. If you found this video helpful, make sure to like, share, and subscribe to the Life Science Coach for more educational life science content. Also, comment below and let us know which topic you would like to learn next. Thank you for your support and see you in the next video.